Okay. So, good afternoon, everybody. Good morning, Professor Ünlü. We are very happy to host you for this webinar organized by the IEEE Pavia student branch. We are hosting Professor Ünlü from Boston University. His webinar is titled Virus Counter, Rapid and Sensitive Diagnostics Based on Digital Detection of Individual Pathogens. Few words about our guest. Professor Ünlü is a distinguished professor of engineering at Boston University. His research interests are in nanophotonics and biophotonics, focusing on development of biological detection and imaging techniques, particularly in high-throughput digital biosensor based on detection of individual nanoparticles and viruses. Professor Ünlü has received several awards in his career, and in 2017 he has been elevated to IEEE Fellow for his contribution to optoelectronic devices, and in 2017 to HOSA Fellow rank for his pioneering contribution in the utilization of optical interference in enhanced photodetectors and biological sensing and imaging. He's also served as the editor-in-chief of the IEEE Journal of Quantum Electronics. So please, Professor Ünlü, when you want, you can share your screen.
Thank you very much, Valentina. If I may ask everybody to turn on their cameras so I can see faces as opposed to a whole bunch of names.
Yeah, okay, please, my student turn on. My students of my class are actually following this webinar. So usually I ask them to keep their camera closed to avoid any problem with the bracelet. But of course, we can try to keep all the videos open, then maybe just close them if we have trouble. So please, I ask my students to open.
Turn them on for like 30 seconds so we can turn it off. So I get to see faces that I'm not talking to a bunch of names.
I know. That's really a problem in this day to know. I'm giving lecture every day in this way.
Cool. Now I see a bunch of faces. That's good. Well, good afternoon. I guess most of you are in Italy. Yes, or all of you are, except for Elisa. I think all of you are in Italy. I know Elisa is in the lab. That's why she can't be in Italy at the same time. So I'm going to share my screen.
Yes, go ahead, please.
And I'll try to give a talk for about 30-35 minutes.
Okay.
So thank you very much for the invitation. It's a pleasure to be giving this talk. And as Valentina mentioned, the title is Virus Counter. And how we're doing this is by detection of individual pathogens. So let me move things around a little bit so it doesn't interfere with my screen. And the technology I will describe to you is easy to understand because this is a phenomenon you see every day when you look at soap bubbles or on a rainy day at a gas station. You can see rainbow colors when you look down with oil film on top of the water layer on the ground. You will see these beautiful colors. And this is due to light interference. I'm an electrical engineer. I've been trained in electromagnetic theory as an undergraduate student. And then I started to work on semiconductor devices. Finally, about 15 years ago, I got very interested in biosensing in imaging. And it was a very important turning point for me to start working with Marcello in CNR in Milano. And ever since, we've been doing a very good collaborative work, specifically on interferometric sensing. I'm in the Photonics Center at Boston University. I'm also appointed in the physics department, biomedical engineering departments also. So today I will tell you how we can take a simple phenomenon as interference of light and how we observe rainbow colors on soap bubbles and convert that into a high performance virus detection instrument. So the outline is, I will give you a little bit of a background and motivation and tell you about the technology platform and review our preliminary results and what we are doing today. To start my presentation, there are lots of analytes that are indicative of disease. And what we do is we try to detect these analytes in a variety of different solutions. So they can be from small molecules to proteins to cells. It's a very large size scale of these different types of analytes, and the detection modality will have to be different for each one of them. So when we look at the ongoing problem, the pandemic that devastated the whole world, and Italy was one of the places early on, it was in deep trouble, and we are not out of the troubling days yet. There's some light at the end of the tunnel, but it's still devastating the whole world. So if you take something like the SARS virus, there are different ways of detecting this, the presence of this virus. And typically, the detection technologies are looking at some biomarker of this virus, such as either the surface antigen, as in the case of an ELISA test, where you capture the target antigen and you decorate it with a detection antibody and have an enzyme or a fluorescent label, which makes it visible. An extension of an ELISA test is a lateral flow assay, in which case you're using gold nanoparticles to label the captured target proteins, and that appears as a faint red line on the lateral flow assay. The most common technique is looking at the nucleic acid, RNA of these viruses, and benefiting from the biological machinery, polymerase chain reaction, to make many, many copies of this RNA molecule. So typically you make 20 to 30 cycles of amplification. You can imagine you're multiplying whatever you have with 1 million or 1 billion, and then the resulting quantity is easily observable. Over the last 10 years or so, there's been lots of interest in doing single particle detection. And the engineering or science approach to single particle detection has been based on either reducing the volume, so presence of a single particle is substantial difference in that small volume, as in the case of digital PCR or single molecule assay, Simoa, that is commercialized by a company in Massachusetts called Quanterix. Alternative would be using very high quality factor resonators to amplify the optical signal generated by capture of a single particle or a single pathogen. So what we do is different than either one of them. The motivation for virus detection reaches far beyond the current pandemic. When I was studying why we should be working on viruses, I came across some papers describing the amount of abundance of viruses in the biosphere on Earth. So there are more viruses on Earth than there are stars in the whole universe. And also, if you take a sample of the seawater, you have millions of viruses within 1 milliliter. So it is extremely abundant species, and they come in variety of sizes and shapes. You can see the coronavirus is actually in the middle of the size scale. Variola virus is the really gigantic virus, almost as big as a bacterium. And Ebola virus is filamentous. It is only 80 nanometers wide, but it can be a micron or 2 microns long. And this is an electron microscope image of the Ebola virus. And the way it was understood as a different virus compared to the other hemorrhagic fever virus called Marburg virus was under electron microscope, because people were able to take an image of this and say, this is not the same virus. And one of the viruses we work on for developing a variety of different modalities of detection is a bovine virus. It's a vesicular stomatitis virus, VSV, and that is a bullet-shaped virus, the volume of which is similar to coronavirus. So first, I will tell you a little bit about our platform, and I will tell you at the very end, this requires an extremely multidisciplinary approach to develop a otherwise simple looking technology. As I said earlier, our technology is based on light interference, and I have these side-by-side pictures of a soap film and an oxide-coated silicon wafer. Both of these are photographs. So in the soap film, you see colors because light reflects from the top of the film and the bottom of the film, and reflected light waves interfere either constructively or destructively, depending on the specific thickness of the film at the specific location and the wavelength of the light. That interference is either building the reflected light or diminishing the reflecting light. So you see rainbow colors within this visible spectrum of the human eye. The best soap bubble you can make is on a silicon wafer. When you have an oxide coating on it's a glass layer, you can step the thickness of the glass as they have done in this photograph. This is actually done by students in an undergraduate learning environment. So with these steps, you can see beautiful, vibrant colors. And this is an actual photograph. So by looking at the color, you can determine the thickness of oxide within a few nanometers. By using optical instrumentation, we can do that determination with 1,000 times better or so at a picometer accuracy. That corresponds to being able to detect picogram per millimeter square bioaccumulation on the surface. The other advantage of the oxide surface is it can be easily functionalized with capture probes. Glass chemistry is the most familiar chemistry in biological studies. And you can make a microarray of capture probes on the surface and do a multiplex detection. protein microarray chips with hundreds to thousands of probe spots can be easily fabricated on such chips. The significant advantage of using the silicon platform is there is a well-established industry. Many trillions of dollars have been spent over the last 50 years or so in silicon fabrication. And we can fabricate these type of chips very easily using standard silicon processing technologies. And this photograph shows a 150 millimeter wafer with about 42 chips fabricated on a single wafer. So the platform can measure the thickness of the film. Therefore, if biomass is captured on the surface, proteins, nucleic acids, whatever is captured on the surface, regardless of what the analyte is, it will form slowly a film, which is adding to the thickness of the silicon dioxide layer. And we can do a reflection measurement and determine that thickness dynamically as more binding occurs on the surface. And this can be used as a label-free detection technology for acquiring binding affinity curves on rates and off rates of molecules from the solution onto the capture probes on the chip surface in a multiplex format. I'll tell you a little bit more about this later on, but this is a technology we have commercialized. We started to sell this instrument under a company called Iris Kinetics. We've also realized about 10 years ago that one can detect individual biological nanoparticles on the same platform instead of waiting for an ensemble of molecules forming a film. An individual biological nanoparticle has substantial mass, substantial optical signature to generate a signal in the image. In case of proteins and nucleic acids, their optical mass, so to speak, is much smaller. So they will not generate a signal on their own, but we label them with a non-fluorescent tag, such as a gold nanoparticle, specifically in rod-shaped nanoparticles. And then we can do single molecule counting, either for proteins or nucleic acids. In which case, this is going to be outside the scope of our discussion today. We can do single molecule detection in a multiplex format with femtogram per ml protein sensitivity and attomolar nucleic acid sensitivity. Okay, just a few words on what we do with label-free detection. This is a multiplex affinity tool. And this is the project Elisa is specifically working on. A couple of these results are from her publications. So in this case, we are imaging the chip surface under a low magnification optical system. And the images acquired on the camera are converted to height images. And from those height images, you can get multiplex binding curves. And we can do this for small molecular weight analytes all the way to biological nanoparticles, treating them as a film forming on the surface. Because the optical magnification is really low, in particular instrument, we have 2x optical magnification. The lateral resolution of the system is in tens of microns. So therefore, everything blends together and you are measuring the thickness of the film. So from which we can get affinity measurements on rates and off rates of binding. Now I'm going to switch to what we do for single biological particle detection. as soon as people developed the microscope, of course, they were interested in seeing the small things that they didn't know that were causing diseases. So early on, 400 years ago, almost, they were able to see cells and bacteria looking under a very rudimentary, very simple microscope. In reality, the design of the optical microscope has not changed tremendously since then. So it is difficult to see small particles in an optical microscope for the following reason. We are typically using visible light to make these observations. Optical microscopes, light microscopes, refer to using visible light. Unlike electron microscopes or fluorescence microscopes, you are only looking at the contrast you will see with your eyes, effectively. So when the particles are smaller than the wavelength of light, let's say typical, the middle of the visible spectrum is 500 nanometers, and typical virus is about 100 nanometers. So it's 5 times smaller than the wavelength of the light, the contrast mechanism of light scattering. I promised I won't have many more equations, but this is just one equation just shows you that the signal generated by light scattering from a nanoparticle is proportional to square of its volume, square of its mass, effectively, or 6 power of its diameter. When the particle is 10 times smaller, the signal generated by that is 1,000,000 times smaller. So biological nanoparticles under light microscope in a regular glass slide preparation are invisible. They blend in the background and they cannot be seen as individual particles. But if you were to put this reflecting surface underneath, now you are effectively doing an interferometric imaging. It is the same concept as listening to FM radio. We always use in engineering, in communications, interference or heterodyning, homodyning to amplify the weak signal. So this is no different than that. If the scattered light is really weak, we can interfere that with reflected light. Effectively, instead of looking at the square of the light scattered, we're looking at the light scattered directly by mixing it with the reflected light. And the signal now depends on the third power of the dimension. So instead of 6 power, it's depending on third power. Effectively, it can be 1,000 times more visible. And this is the basic principle of being able to see virus-sized nanoparticles on our system. So to prove that this was actually quite simple to implement, when George Daboul was an undergraduate student at Boston University Biomedical Engineering Department, he started to work in my laboratory with zero technical training on optics. I actually had to give them an optical photonics book so they can learn a few of the chapters before they start to work on this. Since then, he's completed his PhD, started a company. Now he is one of the founders of NanoView Biosciences. So the basic idea was when you have reflection from the flat surface, you should see nothing on the image. When there is a particle, light scattered from the particle will interfere with the direct reflection reference signal and will generate a bright or dark dot on the surface. And then if the particle is different sizes, then you can judge the size of it by looking at the brightness of that dot. And that works quite well. And my students, David and George, actually have commercialized this technology for exosome detection. We worked on virus detection for many years, and we collaborated with our medical school. A virologist, John Connor, made significant contributions to the development of this technology. With him, we worked on hemorrhagic fever virus detection. And early on, we used these model viruses that he prepared, these VSV pseudotypes. They are genetically modified to express the glycoproteins of a variety of different hemorrhagic fever viruses, Lassa fever, Marburg, and Ebola. So they are stand-in, they're models for the actual hemorrhagic fever viruses. So the basic idea is you have an antibody array, you image the surface, you don't see any dots corresponding to viruses. So you incubate, and then you, before incubation, you get an image. After incubation, you get another image. And based on the size of the particles you observe in your image, you can determine the number of particles that are consistent with what you expect from viruses to produce. And that allows you to eliminate the background and specifically detect viruses at one virus limitation. So we have done those studies first on incubated, washed, dried images or on chips after drying the chips. Later on, we developed a flow cartridge. And in this case, the chip is inserted into a multi-layer laminate cartridge system. And the detection is effectively one step. The chip has antibodies against the virus. You insert the serum solution or buffer solution, depending on what kind of experiment you're doing. When you close the lure cap, it pushes the solution into the channel. When you release the air seal, air is allowed to enter the chamber. And the wetted, when you push the sample down, it fills the channel and wets the paper suction area effectively. And the paper drawing the liquid through capillary action pulls the liquid through the chamber at a constant speed due to the shape of the paper area. So in this case, you can basically image and start seeing individual viruses, and you can count them. And this is the basic modality of our detection. And it worked quite well. We were able to count the individual viruses. And that allowed us to have about 100 times better sensitivity compared to other rapid techniques, mostly lateral flow assays. And this is a study we did on actual bona fide Ebola viruses. We could not study them in our laboratory at Boston University at the time. Our BSL-4, biosafety level 4 laboratory was not functional yet. So we had to reach out and work with the University of Texas Medical Branch in Galveston. And they did these experiments wearing suits like that. People had to be dressed up like astronauts to run such experiments. And we delivered instruments and chips. They run the experiments, and we showed that single virus counting can be rapid. These were 20-minute tests, and it can be quite sensitive. later on, we tried to improve the sensitivity further. And instead of directly immobilizing antibodies, we wanted to improve this capture efficiency by immobilizing the capture antibodies with flexible tethers. And what we know is DNA can be flexible. It can be specifically captured, immobilized on the surface with an amine modification at the bottom end. And the complementary DNA can be conjugated to the antibody. So antibody is elevated from the surface and it is more flexible. It's allowed to move around to capture the virus. And this also allows you to convert a surface to a specific antibody surface, starting with the same chip for different purposes when necessary. So in this particular case, this data is having a 4-multiplex DNA chip. The first step is you take a soup of different antibodies with DNA tails corresponding to these spots, incubate with the chip, and the chip is now converted to an antibody surface, and then you run Ebola sample, Marburg sample, and Lassa fever sample, and demonstrate that in a multiplex modality, we can detect these viruses without interfering with each other. So this was a very useful study that led to what we are doing now. And that is, instead of converting the surface to an antibody surface, we convert the virus itself with antigens around it to a virus with DNA molecules sticking out from. So basically you incubate or mix the DNA conjugated antibodies with the virus sample and make the virus what I called a hairy virus. And all those hair molecules are DNAs that are complementary to a specific spot on your sample. And we demonstrated, actually that paper was published in 2021 in January. And we demonstrate that the capture efficiency is actually better. And it comes with an additional advantage. And that advantage is your chips are generic chips. You can have a standard chip to use for whatever you want to capture. And the assay is defined by this DNA antibody reagent. We also show that you can freeze dry, lyophilize these molecules, and they have a pretty decent shelf life. So 14 days at 35 degrees C actually corresponds to about 150 days at room temperature. So these things can be kept, the reagents can be kept at room temperature for a long time. And the chips effectively have infinite lifetime. So we published this paper a couple months ago. And in addition to having the advantages of long shelf life and simple assay, it also has better sensitivity. We were able to demonstrate a limit of detection in the order of 40 PFU, plaque forming units per milliliter. The results, this is a Italian paper in Lombardia. They have studied the viral load of patients with symptoms or without symptoms. And the range of viral load was in the order of 10 to the 4 to 10 to the 6 viral particles, excuse me, viral particles per milliliter. And that is derived from the PCR cycle threshold numbers. At the low end, at 10 to the 4, they needed to have 31 cycles of PCR. Even if we have, let's say, 1 to 10 correspondence of viral particles or effectively cell-free copies of the nucleic acids, about 1 to 10, 400 PFU would correspond to less than 1,000 viral particles or RNA molecules per milliliter. In which case, we see that we are compatible with more than 30 cycles of PCR, and we will capture efficiently the viruses and detect them for the vast majority of infected patients with or without symptoms for coronavirus. So this was part of our motivation to move on to what we are doing now. So we are building this now a compact instrument, pivoting the existing technology we were able to package together with automated fluidics, and benefiting from this concept of having DNA chips and reagents in a very simple assay platform. Once again, we have the DNA chips. Those are generic. Regardless of what you're trying to detect, you make the same chip. And then you make a corresponding reagent. And that reagent is the antibody against whatever you're trying to detect, with a DNA tail complementary to one of the spots on your chip. If you wanted to do a 4-plex, then you can have 4 different antibodies with 4 different DNA molecules. In the assay, you mix the sample and you incubate. And let's say you're trying to do a multiplex detection of 4 different targets. And in this case, I'm showing that one of the targets is the bacterium, E. coli, and the other one is the coronavirus. We could combine them in the same, well, you have a target solution. You can add reagents against the coronavirus and other viruses, also E. coli, basically shake the tube and incubate the contents on the chip and start counting. Now, when you have DNA molecules and corresponding antibodies in the solution, if they do not find a biological nanoparticle to bind to, they will still bind to our chip. So in a conventional label-free detection technique, that will generate a enormous background signal. It doesn't generate any signal for us, because a blanket of molecules binding to the chip surface, they do not have dots in the image. They will have the entire surface to get brighter or darker, but they will not have a individually identifiable contrast points on the image. So those free DNA antibodies, they get captured on the surface, but if they have a virus or bacterium with them, then they generate a nice dot signal. So this is what we are doing right now. And we are taking this point of care instrument that we have built for affinity measurements, and we're modifying it to work for virus detection. We do have an NIH grant under the RADx program. And this technology will allow us to do maybe 100 tests a day on an instrument. We also have the same concept implemented on a well plate reader, in which case the chips are inserted into well plate. You can do this with 96 chips, and you read these chips from the bottom through the glass. So there are well plates with glass bottoms, for optical imaging. And in this modality, we can image 96 chips, one after the other on a single instrument, in which case we can actually get to thousands of tests per day. Finally, I want to say a few words about our recent results on bacteria detection, which is connecting to this multiplex multimodality detection platform. We were able to show on the IRIS platform, incubating them in static solutions, we were able to get down to single-digit CFU colony-forming units per milliliter endpoint detection in a 2-hour incubation. We did the studies with E. coli. We're able to get 2 CFU per ml sensitivity. First attempt. It was not very difficult. Now we have modified our system to use it the same way with DNA conjugated antibodies as reagents and DNA chips. And it does work quite well. As you will see in this video, when we capture a bacterium, we know it is a bacterium. You know, there is no ambiguity in terms of what is this that we're detecting. And as you see, these binding curves show discrete steps because we are counting. And the lowest concentration appears to be 10 to the 3 CFU per ml. But you have to understand that this is a 10 microliter solution. What we have is 10 bacteria in the volume, and we can still count individual bacteria and do the detection at that level. And one application area is urinary tract infections, and in which case the clinical relevant concentration is 10 to the 5. I submit to you that at 10 to the 5 CF per ml, within minutes, we have sufficient count of bacteria to have positive diagnosis. So we can discuss the benefits of individual counting of viruses or bacteria. One of the significant differences is we are looking at intact pathogen. We're not looking at a fragment or a signature of there was a pathogen. Maybe there is one, maybe there isn't at the time of detection. We can tell infectivity of the people much more accurately. And we have rapid tests, and it is a linear process, right? The more you have, the quicker you detect. It's not a logarithmic system as in PCR. So in PCR, if your virus concentration is 1,000 times higher, you don't really benefit from an improved speed. If instead of doing 35 cycles of PCR, you may end up doing only 25 cycles of PCR, but you still have to do the RNA extraction, you know, and 25 cycles of PCR. And instead of a 2-hour experiment, you can shave it down to 1 hour and 45 minutes. If our viral content is 1,000 times higher than our limit of detection, we can have an answer at 1% of the time that is required for at the limit of detection. So there is one other interesting application. It's specific to bacteria counting. Since we're imaging, we are watching the bacteria as they get found, captured on the surface. In the presence of a growth solution, they grow, they multiply, and you can see how that is affected by introducing different drugs and determine antibiotic susceptibility. We're working on that. We have very encouraging results, but that would be a topic for another day. At that point, I mean, I should probably show you a picture of the people who made lots of contributions to this work early on. when we worked on this over the last 10 years, variety of different people made contributions, and the disciplines involved are from chemistry to biology to medical to electrical engineering, computer engineering, mechanical engineering, biomedical engineering, material science. It's all represented in these pictures. And this is a more recent picture of our group. And I also want to extend special thanks to Marcella Chiari, not only for being a wonderful collaborator, but also introducing me to Elisa. She's been an extremely productive member of our group, and she is in the audience. She may be blushing a little bit. That's it. So if you have any questions and if you have any time left, I'd be happy to answer.
Thank you very much. And we can, big thanks to Professor Ünlü.
Yes. Thank you so much for the super interesting webinar. So yes, we have time for a few questions.
I need to be able to stop share. Perhaps.
Okay.
Yes.
So is there any question from our student, PhD student? I don't see any.
Hi, everyone. I would like to ask a question. Is more a curiosity, actually. I would like to know if, okay, you talk about the counting of different virus and bacteria together, but it's also possible to count different type of virus instead of a virus and a bacteria. I mean, they have like the same structure, more or less. It's possible also to do that?
Right. The multiplexing is not based on size discrimination. Multiplexing is based on affinity. And you have DNA spots of different sequences of DNA at different locations. You're taking an image and you're looking at this spot versus the other one. And whichever one has the dots on it when you run the solution, that means you have viruses with that DNA attached to them are captured. So the DNA is attached to a specific antibody, and antibodies can be extremely specific to the virus type. So yes, I mean, as we have shown in our work with hemorrhagic fever viruses, we did multiplex detection of pseudotypes for Ebola, Marburg, and Lassa fever on the same chip, side by side.
Okay, thank you.
Basically, physical location of where they are bound is telling us what it is.
Mm-hmm.
Discrimination of the size is a validation point.
Okay.
You know, if other things, some dust particle or something is bound to the surface, it doesn't look like a virus or bacterium, we can eliminate those to improve the fidelity of the measurement.
Mm-hmm. Okay, thank you.
Any other question?
I also have a question myself. So it is often said that the gold standard for biosensing is SPR, so surface plasmon resonance. So I was wondering, you are laughing because I know they are a bit like your enemy. But so I was wondering if you could discuss a bit about the advantages or differences in using the iris system with respect to an SPR system.
Okay, so what you're referring to is the multiplex kinetic measurements, or ensemble measurements. You're getting on rates and off rates of molecules. Well, SPR has the advantage of being the first successful label-free detection method. It came out in 1990s, where the camera technology wasn't nearly as good as today. You could not image a surface and determine the reflectivity of a surface with great precision. But now you all have cameras on your cell phones, which are super cameras. The pictures you take with your camera today on your cell phone is far better than what I could take with a beautiful, single length reflex SLR camera in the 80s. Right? You could buy a $10,000 camera. It would not produce a photograph as good as your iPhone camera today. So technology improves. The reason our technology works well now and better than SPR is the optics, light illumination, and imaging. The CMOS cameras are far better. Our technique is bright field reflection. So just to compare to SPR, SPR is looking behind the wall to see what is binding on the other side of the wall, right? Because there's a gold film, it's opaque, and also it is very difficult to understand. If I were to ask you, all of this group, whatever 30 some people in this meeting, 46 people, if I ask you how SPR works, I bet only very few of you will be able to give me an answer. If I were to ask you, what is the polarization of light for SPR to work? I guess only a fraction of you can answer that question. I've asked one of my advisors who worked on SPR.
It will be the topic of one of our future lectures.
I would love to talk about it. So the basic idea is we can do this by looking at the reflection with a concept as simple as soap bubble. But we could not do this 10 years ago. Since our technique is bright field, so you're looking at light reflecting from the surface, our sensitivity is directly proportional to the square root of number of photons we can bounce off that surface and detect on a camera. And it is the square root. So we are trying to measure the reflectivity change as little as 1 part in 100,000 or 1 part in a million. That means we need 10 to the 11, 10 to the 12 photons bouncing off the surface. And that is entirely feasible today. With the cameras that we have, we can do this, and we have shown that we can do this very precisely. The other significant advantage, and this is perhaps the most significant advantage, is when you do a detection of molecular binding based on local refractive index differences, label-free detection is generally an optical techniques is based on that. You cannot distinguish between local refractive index changes due to solution refractive index versus binding on the surface. SPR, biggest problem is so-called bulk effect. So I can tell you that if you read Biacor's user manuals, they tell you this bulk effect is sometimes referred to as DMSO effect. That's the typical solvent that they use for small molecule samples. 1% DMSO and a Biacor instrument will generate 1400 RU signal, whereas small molecule binding to the surface generates about 40 RU signal. So you have 30 times larger background than your signal. And then you have to go through very accurate referencing and preparation of the solution, so on and so forth. I mean, SPR has been an engineering marvel. Despite all these difficulties, despite starting with a very rough gold surface, because gold, I mean, if you're wearing any gold jewelry, do you have any pure gold jewelry? The answer is no. Gold cannot be polished. It's too soft. It has to be mixed with other things. It's not 24 carats, but all the jewelry is 18 carats or 14 carats for bracelets and rings and so on and so forth. I have a ring here. So, but for SPR to work, you need to have pure gold. So it's rough. And the sensitivity is not good enough, so they have to put this dextran layer to have the molecules bind at many, many layers. And that even complicates further the dependence on local pH, temperature, salt concentration, whatever else. We published a paper a month ago, and Elisa was one of the authors, where we showed on our system, we can eliminate the bulk effect completely. Because the signal due to surface binding versus signal coming from solution changes are separable from one another. I don't think there are any other optical detection techniques for label-free detection that can claim the same thing. SBI should not exist for too long. Ours is easier to understand, easier to use, and produces better results. So we'll see.
Thank you.
Are there any other questions? I have myself a question. Do you need, what kind of sources are you mainly use right now? LEDs or particular sources?
Well, we started out with a tunable laser long time ago, extremely naively, about 15 years ago. And we quickly moved to using LEDs. That's another thing. If you go back 14 years ago, 15 years ago, LEDs were hard to find. Now we all have LED light bulbs, right? So LEDs are very robust, readily available. We use different color LEDs. What we argue in the bulk effect free measurements, you don't even need to know the oxide thickness precisely on your chip to operate label free by mixing the colors of the LEDs to get an effective color that will give you the bulk effect free measurements. So we're using regular LEDs.
Okay, so any other question? No, okay, I don't see any other. So don't be shy. It's a good opportunity to get in touch maybe with Professor Ünlü if you then want to get a PhD at Boston University.
It kind of worked out for Elisa, so.
Yes, and also other previous student, I mean, one of my PhD students, she spent 6 months in his lab. So we already have some connection. So if you are interested and if this research are fascinating for you, you can also try to find a scholarship and go and work with him on these topics.
And this research is interesting to you, you can also walk over to CNR and talk to Marcella. And
because Marcella is not in Milan anymore.
She is CNR. I said.
Okay.
What is the name of this street? Mario something Street.
Oh, okay. Okay.
A block north of Porpora.
Mm-hmm. Yes, yes. Okay, so if there are any other questions, I think we can thank Professor Ünlü.
Thank you.
Yes, thank you so much for your talk.
Thank you for this opportunity to speak to all of you. And if you have any questions, you can just send me an email.
Okay.
Refer to this discussion. So
okay.
You won't be a complete stranger. Take care, guys.
Thank you so much. Bye bye.
Okay, per I miei studenti, for my student, I think we can close the lecture here, so we don't have to go back in our Zoom room. Non è necessario